First speaker today is Dr. Mangold. She's a professor at Montana State University in Extension Agricultural and Natural Resources. She received her bachelor's degree from Iowa State and then a master's and PhD from Montana State. Her research focuses on integrated management of invasive plants on rangelands and natural areas. And in 2011, she was funded through the regional IPM grant program to develop a statewide early detection rapid response program for Montana. One of the interesting parts of that was she included in that program a competition among weed districts in Montana to see who could have the most sightings. When I was reading about that, I thought I wasn't really sure if the winner was really the loser because I wasn't sure if sightings equated to abundance of weeds. Anyway, today Dr. Mangold is going to talk about revegetation efforts following the removal of invasive plants.
Well, thanks a lot for the introduction, Matt. That seems like a long time ago that we were doing that work, and I'm glad to see Peter Rice is on the webinar. He was a co-PI on that project. So today I'm going to talk about something different. I'm going to be talking about revegetation of weed-infested rangeland. And let me know if you can see the full slide version of my screen. All right, I see thumbs up. Well, I'm really happy to have this opportunity, and I'd like to thank the Western IPM Center for doing these webinars and for reaching out so that I could talk about revegetation. This is something that I have dedicated a good portion of my professional career on so far, and there's room for more work. So I'll probably be working on it into the future. So this is just a slide of, this is one photo from an area in Western Montana that it's historically native rangeland that has been invaded by cheatgrass, which has been called, one of the, if not the most invasive plants in the Western US. And in situations like this, when a landscape becomes heavily dominated by an invasive plant, perhaps it's been dominant there for, years to decades. Simply controlling the weed is not enough in a lot of situations. And that's where this idea of revegetation comes into play. So I have on the slide here, there's kind of this cycle that we're going to walk through. We typically see weeds and we treat those weeds. Herbicides are the most common way to control the weed, but there's other ways as well. But we treat the weeds, and when we treat the weeds, it opens up space in the plant community, what plant ecologists like to call niches. And typically what happens is the weeds reestablish and they occupy those niches that have been opened by our weed management activity. So managers treat the weeds again, we open up those niches, and the weeds reestablish. And what revegetation does is it attempts to come in to these systems and deliberately introduce species that we'd like to have growing there. And this is done in situations where, like I alluded to earlier, the weeds have been dominant for such a long time that the remaining remnant vegetation isn't enough anymore to fill these niches on its own once we do control the weeds. So revegetation is a form of cultural control. If we think about kind of the broad categories of integrated pest management, it would be called cultural control. And like I said earlier, it's the deliberate introduction of plants. And the goal is to establish vegetation that competes with the weeds, but at the same time meets our management objectives. So in a rangeland setting, that could be forage production for livestock, it could be wildlife habitat, it could just be aesthetic purposes of having native plant communities that are diverse. Maybe it's supporting pollinators, etc. And in most situations, revegetation is usually done by integrating seeding with some sort of weed control. And it's commonly herbicides because that's the most common way to control weeds in rangeland settings, but it could be something like targeted grazing, prescribed fire, biological control. Mechanical control not used as often in these types of settings. So that's kind of an overview of just why revegetation and what is it. So this is revegetation is if you decide as a manager to revegetate, it's a fairly involved process. And this is just a schematic from an MSU extension publication talking about revegetation. And you'll see there's a lot of different steps that need to be taken into account if you're planning a revegetation, if you're planning revegetation. And it can be pretty complicated. There's a lot of things to think about. I kind of jokingly refer to this as the 12-step program for reducing chemical dependency. Sometimes people laugh at that joke. Sometimes they don't. Thank you, Steve. I can see you laughing a little bit. But you know, all joking aside, revegetation is complicated and it's expensive, especially if you're seeding native species. You can spend $300–400 an acre if you're buying native seed and putting that out. So it's expensive, and unfortunately, it doesn't work more often than it does work, and and that's due to a lot of different things. But one of the critiques of revegetation is that it historically has relied on practices that are typical of agronomic systems. Um, you know, driving tractors with large implements over a landscape that is really, you know, perhaps more variable and more complicated than an agronomic system might be. So what I've been doing along with my colleagues and many other people across the Western US is trying to understand how we might apply plant ecological theories to revegetation practices in an effort to help those practices align more with what's going on naturally on the landscape, and hopefully increase the success of revegetation. And some of the work I'm going to share with you today is really looking at how we have integrated herbicides and seeding in weed infested areas. And I'm going to focus on kind of these two ecological theories, one being a seasonal priority effect, and the second one being plant successional dynamics. And I'll talk first about the seasonal priority effect. So if we think about weedy species, they usually have faster germination rates. And once those species emerge, they grow faster. That's what makes them weeds, right? And this creates what is called a seasonal priority effect. And it's just that the species that emerges first gains an advantage over slower growing species. So if you're in a revegetation situation where you have seedlings trying to compete with seedlings, it's very difficult for the native species to outcompete these weedy species. And this is a photo I often share looking at cheatgrass, which is shown here on the left, competing with bluebunch wheatgrass. There's two little seedlings there shown on the right in that blue circle. And this picture was taken in the spring, and the seeds that produced both the cheatgrass seedling to the left and this bluebunch wheatgrass seedling to the right were seeded on the same day the previous fall. And you can see that just naturally, because of their biology, the cheatgrass has a competitive advantage. And with revegetation, the typical scenario is seeding in the fall. You might apply herbicides in the fall or maybe the previous spring or summer to control the weeds, and then you would seed your native species, oftentimes grasses, into this scenario. So by the time spring comes around, like with the cheatgrass, bluebunch wheatgrass scenario, the cheatgrass is way bigger than what the bluebunch wheatgrass seedlings are. So that's the typical scenario. So really over the last 10 years or so, working with a lot of different people at Montana State, we did a series of studies that we looked at this seasonal priority effect idea, and we wondered if there would be, first we wondered if it really made a difference with cheatgrass competing with bluebunch wheatgrass. And then we wondered, well, could we test it in the field and think about strategically timing herbicides and seeding to create a seasonal priority effect for the native grass, bluebunch wheatgrass. So we first started a greenhouse study where we seeded cheatgrass and bluebunch wheatgrass. We either seeded them together at the same time, or we seeded bluebunch wheatgrass 2 weeks or 4 weeks ahead of cheatgrass, and then we looked at the competitive dynamics. And what we found was that the early bird did get the worm. When the two species were seeded at the same time, a 10-fold increase in cheatgrass density decreased bluebunch wheatgrass by almost 60%. When we seeded bluebunch wheatgrass like 2 weeks to 4 weeks earlier than cheatgrass, that same increase in cheatgrass density only decreased bluebunch wheatgrass biomass by 13%. So there was still competition going on, but it was not nearly as extreme. And that picture on the right, this is bluebunch wheatgrass, the taller species, the taller grass that was sowed about 4 weeks before the cheatgrass down here at the bottom of the pot. The pot on the left is when the species were sown at the same time, and most of that biomass you're seeing is cheatgrass. So it worked in the greenhouse. We demonstrated that we could shift the competition dynamics. So the next thing we did was, well, let's take this out in the field and see if there's a difference in bluebunch wheatgrass if we seed it in the fall versus if we seed it in the spring. So we found that you could seed bluebunch in the fall, which is typical, or anytime in the spring, and they worked equally well in terms of getting bluebunch established. And then we seeded cheatgrass into these plots, and we also found that these big, robust bluebunch plants that had been growing for a year were capable of suppressing cheatgrass that was introduced into the plots. So that was in a fallow field study, really with very limited competition from weedy species, also a very nice seed bed where we seeded our bluebunch wheatgrass. So the final test of this idea was to take it to a cheatgrass infested field. And this photo on the right is showing part of one of our sites. The cheatgrass is all that brown grass growing here, but we also had a lot of western salsify or Tragopogon dubius, which is a weedy forb. So at 3 sites in western Montana that looked somewhat like this, we sprayed glyphosate in the fall or the spring to control the cheatgrass and other weedy species. And then we seeded bluebunch wheatgrass in the fall right after spraying, or in the spring right after spraying. And we looked to see how that worked out. And what we found is that spring spraying plus spring seeding resulted in the greatest bluebunch wheatgrass. And this is from one of our sites looking at the cover of bluebunch wheatgrass. Our different seeding dates are listed on the x-axis here, fall, and then we tried 4 different spring dates. And then the open bars are if we sprayed in the fall and then seeded. The closed bars are if we sprayed in the spring and then seeded. And you can see that the cover was greater when we seeded in the spring and when we combined that with, we did that seeding after a spring application of glyphosate. Here's a photo of our plots. This was, I think this was the first, no, this was the second year after, no, the first year after seeding. And this is all bluebunch, kind of that bluish green grass that's growing here. We drill seeded it so you can kind of see the drill rows. So through this series of research, these research projects, kind of exploring whether we could create a seasonal priority effect for the desired native grass, we came up with this management recommendation. A spring application of glyphosate to control the cheatgrass seedlings, followed by a seeding of bluebunch wheatgrass immediately afterwards. And that allowed bluebunch wheatgrass to grow for 1 full summer before the next cheatgrass cohort emerged in the fall. As a winter annual, that's how cheatgrass grows. And in so doing, we provided bluebunch wheatgrass with this seasonal priority effect, thus reducing its competitive the competitive effect of cheatgrass in allowing the bluebunch to establish better. So that's one example of ecological theory and how we've been trying to um, develop and apply that ecological theory in just something as simple as strategically timing the way we integrate different weed management control or weed management strategies. The other concept I'd just like to share in the last 5 minutes or so is this idea of plant successional dynamics. We know plant communities change through time. And I've been really fortunate in my time here at MSU to have a couple different opportunities to look at the long-term outcomes of revegetation. Revegetation is not a quick fix. These landscapes have degraded over many, many years in most cases. And in order to restore them, we also have to kind of take a long-term approach and really consider plant successional dynamics. So one of the opportunities that I've had here at MSU is to look at some studies that were conducted again in southwestern Montana. These were studies that were implemented in the early 90s where different spotted knapweed control measures were integrated with seeding. And thankfully, some of the people involved in this initial research were still around. They still had plot maps of these sites and the details of what was done at these sites, along with the papers being published, so that we went back to these sites 15 years after they were first seeded and sampled them again. And I'm only have time to kind of give you the high points here, but the seeded grass reduced the invader. And here you can see a strip of bluebunch wheatgrass to the right of this black line that was seeded, as opposed to a strip to the left of it that was not seeded, and it's still dominated by spotted knapweed. A little bit of the data, looking at the seeded grass biomass. This is data from year 2. This is across different seeding rates. That was one of the factors that was manipulated in one of the studies. This is 2 years after seeding, 6 years after seeding in the middle, and then 15 years after seeding to the right. And what we found was that seeded grass biomass increased from about 26 pounds per acre in year 2, so if averaged across these, to about 1760 pounds per acre in year 15. And at the same time, the spotted knapweed is declining. And this highest seeding rate 15 years later had decreased spotted knapweed biomass by about 86%. Here's just a couple more pictures to show you. Non-seeded, this is a strip of intermediate wheatgrass, a non-native grass that was used in some of the studies, and then a strip of bluebunch wheatgrass, a native grass used in some of the studies. We also found in this study that integrated management increased seeding success. In this case, picloram had been integrated with seeding. And what we found was that seeded grasses produced more biomass where picloram had been applied. And this was more so 2 years after seeding than it was long term. But there was still an effect of that integration of methods even 15 years later. The last study I'll show is just, this was a site where leafy spurge was infesting rangeland, again in western Montana. This site was treated in fall of 2002, where they integrated either 2 different herbicides or no herbicide with seeding of one of 5 different grasses. And these sites were sampled by the initial researchers in 2003 and 2005. We came back in 2016. This was a great example of a group of people coming together. Peter Rice was one of the people that brought his crew down, but lots of people were interested in this project and came out and helped us sample it again in 2016 so we could get these data to look at the long-term effects. In summary, the seeded grasses, they did increase over time, especially bluebunch wheatgrass. That's kind of been a shining star in our revegetation projects. You can see it growing here. And leafy spurge decreased over time. even in the non-treated controls. So it was decreasing regardless of anything we did. So that was interesting. But what was most interesting to me was what the other species did at this site. So here's a picture of the site in 2016. You can see there's a lot of vegetation growing there that isn't leafy spurge and isn't necessarily what we seeded. But what we found, that was a control plot, but what we found was that seeding grasses, especially bluebunch wheatgrass, decreased the biomass of other species. So here we're looking at other biomass, the dark blue bars here that I've highlighted in red, that's bluebunch, that's the other biomass where bluebunch wheatgrass was seeded. And you could compare that to kind of this royal blue color where no seeding had occurred. So what were those other species? Well, those other species were what most people in Montana managing rangeland would consider undesirable. Cheatgrass, Japanese brome, bulbous bluegrass, and some weedy forbs. And these might be what we call secondary invaders. So I think circling back to this idea of why revegetation as part of an integrated weed management plan, if we can get things established that are desirable and they're competitive, we can reduce not only the target weedy species that we're interested in, for example, cheatgrass, spotted knapweed, leafy spurge, but we can also limit the invasion of these secondary invaders, as that leafy spurge project showed you, over the long term. Plant communities change over time, and we need to be thinking about how our management is affecting the plant community dynamics, not just in the short term, but in the long term as well. So I will end with that. There's my contact information. I do want to recognize some of the different groups that have been part of the research that I shared with you today. Much of this research was supported by the Montana Department of Ag Noxious Weed Trust Fund, ARS, NEFA helps to support my position, the Missoula County Weed District and Extension, and NRCS has been part of these projects as well. So with that, I guess I'll stop sharing because I think I can see the chat box a little better if I do that. And then,
So we do have a question in the chat box from
all
Steve Young.
right. All right. Hi, Steve. Do you think the lack of success in revegetation has to do with the fact that we don't like the weeds? Sort of like your agronomic reference. What if we treated them like weeds?
Do you want me
So,
to read the question?
yeah, well, I can read it. I'm not sure I
Yeah.
understand it all, Steve. What if we treat rangelands like a cornfield, but we don't also treat our native desirables like corn? What if we treated them like weeds?
Yeah, so I can just talk.
Okay.
It's probably easier. But I thought, I mean, so right, so we do a lot of work on rangelands, like you said, and Jane, I really appreciate your talk. It was really excellent, and I've seen it before, and I think you're really doing some wonderful work. But this idea of, if we're treating our rangelands like you said, so we're treating it like kind of like an agronomic field, but we're also treating the things that we plant the same way. And if we reversed our thinking about, like, okay, the weeds are there and they're very successful. So why are those weeds so successful? And a lot of times it has to do with they're pumping out seed all the time. I mean, cheatgrass, any of those ones you named, they're producing a lot of seed. And even like leafy spurge has got this root system that goes crazy and it does all this stuff. So can we kind of mimic that with our natives? I mean, typically, like you said, so we go out, we take a drill seed out and we plant rows. And I do the same thing. And I see kind of, okay, we see these little rows of beautiful grass, but is there a way to kind of overwhelm the system or kind of tip it back to the natives or the desirables, kind of give them that advantage that they're not getting, and are thinking, if we're like, how does a weed do it? So can we kind of mimic that with the natives and desirables? I don't know if that's possible, but it's just kind of my thinking, and I'm wondering if that would be more successful that we could have.
Yeah, I think I get what you're saying, Steve, and yeah, I agree. I mean, could we try to could we do things maybe with like seeding rate, right? I mean, let's seed even more seeds than we might typically seed in a revegetation scenario. And that's been tested. Some of that spotted knapweed work, there were different seeding rates in that project. I guess I would challenge us to maybe look at the other side of the coin here, in that our native species tend to do better in stressful environments maybe than our weedy species do. Maybe they don't produce as much seeds, but they have long-lived roots and long-lived individuals as opposed to our weedy species. I think we perhaps need to look more at how we can return some of these disturbed sites to a more native environment, and that goes all the way from what's going on in the soils with microbial communities and nutrient availability, all the way up to what are our grazing systems looking like. So I guess I would challenge us to kind of look at the other side of that coin if we can, and let's try to create conditions that are more conducive for these long-lived, slow-growing, kind of stress-tolerant species, which are typical of our native species on rangeland.
Yeah. We do have two more questions in the chat. The first comes from Brittany. Do you have any data on how the treated and untreated plots in your studies responded to wildfire? For example, were the treated plots more resilient to invasion following wildfire?
Yeah, good question, Brittany. No, neither of these sites where the long-term work has happened, they've never had like a naturally occurring wildfire go through them. It would be interesting to do some prescribed burning and see how they respond, though.
Yeah. And one last question from Len Coop. Did you count or take note of the relative abundance of herbivores such as grasshoppers or biocontrol agents in the plots?
Yeah, that's a really good question too, Leonard. It's both of those long-term sites, the spotted knapweed and the leafy spurge site, they do have established biological controls. Um, we didn't quantify what's out there, but we know there's biological control agents out there. So we suspect that they're still playing a role in the dynamics, probably especially at the leafy spurge site where we saw a decline in leafy spurge. There's also been, those sites have been excluded from grazing. Um,
Okay.
but I think one thing that's interesting is that spotted knapweed site, where there are a variety of spotted knapweed biological control agents, if you look at the sites that weren't seeded, they're still dominated by spotted knapweed. So even though the agents are there, they may be creating smaller, less competitive spotted knapweed, but the spotted knapweed's not going away completely. And I guess that's another argument for why integrating revegetation into the system could help the long-term dynamics.
Great. Thank you very much.
You're welcome.
Uh,
Yeah.
that was terrific. So now we're just going to switch gears and we're going to hear from Dr. Crowe, who is the National Policy Manager for USDA. Today, Dr. Crowe is going to talk about the CAPS program.
Well, just to get right off the bat, I am not a PhD, so I only pretend to be one, I guess. So it's just get that right out of the box. I am going to share my screen though, nonetheless, and we're going to talk a little bit about the CAPS program and kind of where we sit with the APHIS in terms of invasive species detection and early detection. Okay, well that's not working. Let's see here. There we go. Sorry about that. So is that sharing the full screen? It's hard to see. I'm on my laptop and it's hard to tell.
It does not look like it's in presentation mode.
Okay, there. So
Now
now
it
it
is.
is.
Yep.
Great, thank you. So as mentioned, I'm John Crowe, the National Policy Manager, and I sit in the Pest Surveillance and Emergency Management section of APHIS at the policy unit. And I am specifically working in emergency and domestic programs. And one of the programs I cover is the CAPS, which is a Cooperative Agriculture Pest Survey. And I wanted to talk a little bit about surveillance and kind of what that looks like from an international perspective. So ISPM 6, which is the International Standard for Phytosanitary Management, number 6. So there's a number of these international phytosanitary measures that are adopted. This one is for surveillance, and the two types of surveillance systems that they utilize at the international level are the general surveillance and the specific surveys. So thinking back to Jane's talk, some of the information that would have been considered general surveillance is that older data that she used from 15 years prior, looking at historical records and things like that. And that information is accurate, as Jane talked about a little bit in her presentation. But it has to be from a reliable source ultimately, is what the standard says. When we think about specific surveys, kind of mincing words here a little bit for the international world, it's obtained about specific sites and or a defined period of time. So if we think about something like the potato cyst nematode, that's going to be in a specific site and over a specific period of time that we're doing those surveys. So kind of the difference being that historically, let's say potato cyst nematode is known to occur in Europe versus specifically we know it's in a specific region in Idaho. What the CAPS program really focuses on is this specific surveys, and this is a critical piece that we really want to think about as we move forward with the APHIS survey specific to CAPS. And key to this is that the information is verified so that it's not somebody that's not able to correctly identify the host or the commodity or the pest itself. Oftentimes we think about plant pathogens being difficult to identify, and even things like slugs or snails. And we'll talk a little bit more about what a plant pest is. So thinking a little bit about where we are in the spectrum, we're over CAPS really is on this preparedness, this red piece here, and moving it towards the response piece. So kind of it straddles the preparedness as well as the response piece of this safeguarding continuum. And as we kind of consider the rest of the continuum here, that response phase is that initial response that we do to understand distribution and or other things. So if we, again, trying to think about pests like potato cyst nematode, we would be in the response phase for the potato cyst nematode. And then as we are recovering from that are things that we would be taking off of quarantine or perhaps issuing the biocontrol agent. There was some talk about leafy spurge biocontrol. That's really kind of in the recovery phase. And Jane really talked about that prevention phase, moving from recovery into prevention is really where she focused her talk. So we're going to talk on the other end of that spectrum today. There are three general types of surveys that we deal with in the CAPS world in pest surveillance. The first is the detection, which is the kind of finding a pest in the environment or commodity, whatever that might be. And the second is the delimiting survey. And that delimiting survey really defines where the pest is in the immediate area surrounding the initial detection. And then the monitoring survey are things that would happen after treatment occurred. So this would be a lot like what Jane was doing in her talk, which would be monitoring after treatments happened in fields and things like that. But what we're talking about with the CAPS are really that detection survey. Looking for things that are not known to occur in the US is critical to the program and key to where we are on that spectrum for us. So as with pest surveillance and APHIS, we're really looking at a multi-pronged strategy that looks at a structured and transparent assessment process to identify pest threats. So we really try to focus the threats that are on the horizon and figure out what's going to be the biggest risk to us and what is not going to be the biggest risk to us. We develop scientifically sound diagnostics and survey protocols, so making sure that if we're looking for insects, the trap and the lure are accurately identified. Similarly, with pathogen, making sure that the DNA or whatever mechanism we're going to use to identify is correct. And then we make sure that those traps and lures are correct for those diagnostics, whether that is a plant diagnostic or insect diagnostic or something else, making sure that those survey materials are important or are provided, are critical. And then we actually provide the resources to conduct the survey. So that if you are as attached to your state departments of agriculture, this is the state survey coordinator position, or the pest survey specialist in the APHIS world would be the ones coordinating these types of surveys in the states. And the other thing that we're working really hard on is timely reporting of pest survey results. You can imagine with a variety of samples taken, it's very easy to take those samples, but if you don't have the processing capacity, it's really difficult to understand what's going on. So really trying to strike that balance between going out there and doing that field work and then processing in the lab. I think everybody could attest to that. Being the slow part is the laboratory and the number crunching. And then the other last piece of this is ensuring that that information is valid and highly quality and verifiable, making sure that we're not out there just saying something is there, that it's reputable sources that are saying such things. And then the notification, kind of notifying the end, is the last step of this slide. So there are two primary funding sources for pest surveillance. The first is our pest detection line item, which comes directly from Congress. It's an allocated fund. And then you see an abbreviation here, PPA 772, for the Plant Protection Act. And section 7721 really gives us the authority to draw down funds without congressional approval, that it's enacted as through Congress, that we have these $75 million in funding available, and another 25 or 26 million from pest detection, which is allocated annually from Congress. And this money really goes out into several different cooperative agreements to the state departments of agriculture, university, get some portion of both of these funds, as well as other cooperators like our Indian tribes, and some landholders might get some funding as well. And then also a lot of funding goes to our state PPQ offices, making sure that those offices have the resources so that if there is a new pest detection, that we're able to respond to that effectively. So thinking back to a comment that I had mentioned earlier regarding the priority pests, making sure that we're looking for the right thing. This is kind of related to that. Our national priority pest list are those pests that are considered national in scope, which are, I mean, we're thinking about things that are on the horizon, like Tuta absoluta, perhaps, or things that would have broad impact across a very important commodity for us. And we target those specific insects with identities, the threats to agriculture and the environment. So not just looking at agriculture, but the environment as well, thinking about the forest pests that we would look at as well. And then it's regularly revised. We look at these annually, and really our science and tech unit does this for us regularly so that we're targeting the right things at the right time. And so just thinking about what a pest is, the pests are injurious to plants by definition. That's in our the Plant Protection Act that I mentioned in the previous slide. And those could be anything for insects, pathogens, mollusks, weeds. And the National Priority Pest List is available at this link. And again, the 2021 is there. It will be updated. Again, just knowing that kind of what's going on with a few pathogens and pests that are coming in Canada and Mexico recently. We were updating that in the spring, trying to get those pests that are proximal to us identified as best we can. So one of the things that happens when a pest is put on the National Priority Pest List, PPQ provides two primary things: the scientific support, which is the impact analysis that I talked about, making sure that we think that it's going to be a pest here, and then the survey methodologies, making sure we understand how to survey for the pest. And you'll see a picture here to the right is our warehouse with all of our traps and lures that we provide to support those primarily insect pests, but we do support some of the pathogen testing as well. So you'll see about a 10 to 20,000 square foot warehouse down here with 400 to 500 pallets worth of traps and lures in it that we distribute annually. So the approved methods that we'll talk about here in a second is a resource that provides science-based surveys and details on national and state surveys. So it directs the states on how to do the surveys for these pests. It also targets a specific plant pest on the national priority pest list that are threat to U.S. agriculture and or environment. Primarily, the pests are not known to be distributed in the U.S. But if we think about a pest like spotted lanternfly that has limited distribution, you'll see that pest on it, so that our western states that are still looking to be protected and with early detection are able to survey for that as well. We also group them for efficiency. So if we think about going to a cornfield, for example, we would want to look for a series of corn pests rather than just one corn pest as far as a survey, because the trip out there is actually more expensive than the actual survey equipment. Thinking the traps and lures cost, a couple dollars each versus a trip out there, which is, $15 an hour just for the wage of a technician, perhaps. So some of the commodities and the taxon groups. So similarly, when we try to group them, we group them by commodity, like I mentioned with the corn, but we also group them by the taxon. So thinking Asian defoliators, we can probably go into a forest environment and identify a series of defoliators from forest crops from Asia. Cyst nematodes, similar, we'd be able to take one single core or root sample and be able to do a series of nematode tests on those. With the survey supplies, this is near and dear to my heart, so apologize for that. This is one of my other programs that I deal with. We have a national warehouse down here in the Rio Grande Valley where I am right now. It provides a single point ordering system for all the traps and lures to support those insect surveys and some plant pathogen surveys as well. That offer consistent products and statements of work so that you make sure that that glue that you're looking for on your insect traps is the same across the board as is the pheromone. And we also have an extensive quality assurance program that happens. If anybody has done entomology work and those paper boards fall apart on you, that is something that is never pleasurable and never advisable either. So we make sure that the paperboard, the glue, the pheromone are all meeting standards and specifications as part of this program as well. The surveys emphasize on new pests, like I mentioned earlier. The pests not known to occur in the U.S. are considered new for us, and multi-pest surveys as well. So we want to make sure that these not known to occur pests in the U.S. are being streamlined into efficiency so that these high priority pests are identified quickly by using multiple pests through the survey. We're included in the national CAPS priority pest list, so making sure that we're doing what we're saying we're doing, so that when Congress asks, we're able to tell them, yes, we are looking for these pests, and we did look at these in these areas. It just helps with the accountability for everybody and increases funding long-term in theory. It has not happened in practice, but in theory it sounds great. May include pests of importance to state as well, so making sure that if the state has a corn pest that's of concern in their state, but maybe we're not concerned about it because it's an endemic pest, but maybe out west, but Illinois is interested in the Midwest, they can go ahead and add that corn pest to their survey if they choose to do corn and they add a priority pest. So it's not limited to only the national priority pests. And I want to just emphasize a key point here that negative data is absolutely critical for our trading partners, that absence is just as important as presence for our pets. And as we move through the consideration of absence and presence, I just want for everybody here to think about what that means for our trading partners in particular. That if we show that we do not have these pests not known to occur in the U.S. and we substantiate that, it opens up a potential trade pathway that we may have been closed previously if that data wasn't available to us. So the definition of negative data is valid negative data based on program and guiding principles. So thinking about those approved methods, which could be simple as looking for it on a visual survey, or as complicated as a DNA test for PCR. They are active surveys. Absence and positive does not necessarily mean negative. So if that makes sense, it's kind of a logic problem if you do. So an active survey, presence of a positive does not necessarily mean, absence of a positive does not necessarily mean negative is kind of a critical concept of that as well, meaning that we have to acknowledge that our survey practices are only so efficient at collecting data, and that the percentage of not finding it is sometimes equal to the percentage of finding it, even though we try to mitigate the not finding it part. And so to have negative valid data, it has to target a specific pest. Thinking about a lure and a trap for an insect would have to target a specific pest, or a pathogen would target a specific host. It would also conduct a survey using the approved methods, which the link is available here for all of those things. So feel free to access that. An examination by a qualified identifier. So for APHIS terms, if it's new to the US, we do want to have our national experts identifying it. We've had a couple cases where university professors have tried to do the identification and unsuccessfully created, or actually successfully created, some trade implications for us. So we definitely want to make sure that the APHIS specialist at the Smithsonian is looking at that, and everybody is on board with that actual identification before we start to respond to any type of pests. If the target pest is not present in the sample after examination by the identifier, the method used to capture the pest, it will detect, and that's the point in time that we would do that. So it would be this year we surveyed for this pest and this location is what we're saying as far as our data here. So looking at a few resources that are available here, the CAPS resource page is an excellent page if you're looking for some resources on how to look for or what to look for invasive insects. The approved methods are all here. So these are all the APHIS priority pests. You'll see on the left side are the scientific name, in the middle is the more common names. And then kind of the targeted host lists are to the right of that, as well as the survey methodologies that are used, whether that's a trap, lure, or visual is there. And then the version, as I said, we update this regularly. So you'll see some older versions here, but those are more stable pests that aren't having a lot of success in trapping methods or traps and lure options available to them. Looking at a pest that has had some work done on it, so Adoxophes oreana or summerfruit tortrix is one that's a high priority target for us. You'll see it lists the various commodity guides that are there, as well as the method that we use for the trap and the lure. And then the pheromone components are at the very bottom of that. So you'll see that we actually make this in our laboratory in Otis. So we make that in-house. All of these, a lot of our pheromones for LEPs are made in-house, but in this case it is made in-house. And so Purdue really manages this for us with NAPIS and our approved methods and CAPS resource collaboration page. So shout out to Purdue for them and Cirrus in particular for us. That cooperative agreement does provide support for NAPIS, which has over 3 million records for survey results over the years since CAPS had formed. So I think that actually is closer to 5 million, but they say 3.2. Whatever, we could agree to disagree on that. The data is state-owned and it's restricted access and is required by role. So if you do want some of this data, happy to help you get as much as you can, but it is a little bit restricted. So NAPIS contains survey data on it. I know I'm running short on time here, so we'll try to speed this up. Survey results, it is not distribution, just making sure that that's clear to everybody here. And it provides information about that survey point in time, national repository for that historical data. The negative data is validated, as we talked about, and negative data checks back to the correctness of the data. And so thinking about the public site of our NAPIS site, this is Pest Tracker. So if you are interested in just knowing if something has been surveyed in your state, you can go here and look for those details. So looking at our example of summerfruit tortrix, we would see that in 2019, this was last year's data, we do have quite a few counties that are summarized here for survey data for summerfruit tortrix. So if you're interested in working with us on perhaps receiving some money to do surveys, you can work for your state departments of agriculture and consider putting in the PPA suggestion for some survey work. If you know you're looking for, if you know people looking for jobs to do survey work, always happy to point you in the right direction for that. We're always looking for trappers to help us with that. And I could definitely put you in contact with your state survey coordinator or your pest survey specialist to know if there are summer jobs available for that type of work. With that, I think there is time for questions.
Yeah, John, we definitely have time for questions. Oh, here we go. Let's see. So here's your first question from Jane. How much conversation or discussion occurs between the U.S. and a pest's country of origin? So, for instance, to learn about life cycle, habitat preferences, how to find the pest, and other things. And then there is a second question if you want me to wait and ask that one second.
So sure. So a lot of the trade discussions that happen between the U.S. and trading partners are very formalized. So they happen in bilateral settings where we have a question about this pest and that pest. So for example, thinking about Ralstonia race 3 biovar 2 that was just detected and eradicated in the U.S. We went to Guatemala and worked with them very closely on making sure that that clean nursery stock was coming in, and it was very clear with Guatemala what those restrictions are and need to be in order to clean up production practices there, so that we get clean stock of the different plants that are plant cuttings that are coming in on that. So that could be very formal and but it could also be very informal, thinking about the relationship we have with the Canadian Food Inspection Agency. I don't really, my counterparts up there, I provided traps and lures to them many times where they were out of them, or they've never provided to us, but we've always provided to them. So it can be a very cool experience. So it really depends on the severity of the trade of the pest when we think about that.
Yeah, so there's a second question, and that is there discussion or collaboration between the U.S. and other countries where the pest is pesty for surveying? I suppose that means hard to survey.
Yeah, so we definitely have, there's just some goal 3 for the PPA looks specifically at the ability for us method development for that. So making sure that we have survey and detection methods that are available specifically for the pests that are not present in the United States or may have a limited biography of work done on it. One thing that's interesting that we're finding is that the actual top indicator for pestiness on it, invasiveness of a pest, is really about the number of research articles in its origin countries that it's present. So if we think about a pest that we gave to somebody like Solidago canadensis to Japan, perhaps, there's quite a lot of work done on that Solidago or asters in North America. And then thinking about that kind of relationship to Japan is important. But similarly, that type of work that's done overseas, thinking about an insect like Old World bollworm, we know that a lot of work is done on Old World bollworm, and we don't have it here. And so we're really trying to make sure that the number of articles is in the consideration for that actual pestiness index.
Yeah, great. So here's another question from Len Coop. This one is, do you test new pheromones for those pests that are coming in the host countries as part of your as part of a quality assurance step?
Yeah, so this is a great question for a number of reasons. So the pheromones can vary for the specific insects based on where the regionality of them. For example, going back to Old World bollworm, there are specific blends of Old World bollworm that are more effective or less effective in certain areas of the country. So what we really try to do is hone our pheromones that we produce in-house into more general categories for it, so that it's more likely to capture them, because we're not sure if it's going to be coming in from South America or South Asia or Europe perhaps. And so that's a really tricky question, but we do test new pheromones in foreign countries. So there is some foreign exploration, thinking about our monocamus lures and stuff like that. We definitely look in China for those and work with a lot of collaborators to be able to identify new pheromones offshore as well as onshore. We have some colonies that we work with a little bit throughout the U.S.
Yeah. So I'm not seeing any other questions in the chat. I do have a question about logistics. Presumably, most of these things you're testing for aren't widespread in the U.S., and you don't really know exactly where they're going to be. So, how do you plan on how many, testing modules you might need across, say, a wide swath of country?
Yeah, so that's a whole logistics question there. So we distribute to about 400 different field offices for our traps and lures from the Survey Supply Program, and we distribute roughly 2 million pieces of equipment. So whether that's a trap insert or a trap itself or pheromone, it's quite a large number. So calculating that, and when you get such large numbers of those, is actually quite easy, because you just, if you know, like, you're going to have X number large, and you're going to go X minus a little bit or X plus a little bit each year, it turns out pretty good. And so the largest indicator is the number of dollars going out for the number of components that we use. And what those specific components are relatively small metrics of those 2 million pieces. So it's across a wide area, you're able to average it out pretty easily. It's a little bit, I'm making it sound a little easier than it is, but it's a matter of number of people and size helps equal it out.
Great, thanks. Here we go. So here's another question for you. Do you use software to quantify imperfect detection? As you mentioned, saying something is absent can be tricky because you may just have missed it.
Right. So that's it. We really are working towards a system to have some type of percent confidence in our survey data. We're not there yet. It's going to take quite a lot of resources to get a percent confidence on a specific pest or a specific area for that to happen. So we're not there yet. And so the answer is no, we don't have software for that. We are working towards some modeling software that will help us with that and be able to help on that. And that is kind of the next phase that we're working to start implementation on next in 2021 with that. So PestCast is a system that we're using if you're looking for specifics on that. So that's where we're going with it. We're not quite there yet, but it is on the horizon.
Okay, let's see. I think that might be it for questions. I'm not seeing any new questions arising. So thanks, John. That was terrific.
Yes, thanks, John and Jane. These were both good presentations, and everyone for being on the call today.
So let's see. Our next IPM hour is going to be in December.
That is correct. December 9th.
December 9th. And look for the newsletter to see who the speakers are going to be on that day. The newsletter will come out. It'll be the first Wednesday.
Yeah, it'll be on the 2nd.
Yep, on the 2nd of December. Thank you all for joining.
Thanks, everyone.